Hey guys, welcome to the third video in our series. In the previous videos, we have discussed the purpose of synthetic biology, as well as a method of synthetic biology called antibiotic resistance. In this video, we will be discussing another method, designing plasmids. So what are plasmids? Plasmids are circular pieces of DNA that look like this. In the video, we will be focusing on the interested gene and the restriction site. Plasmids are useful for synthetic biology because they have the ability to replicate independent of chromosome DNA in the nucleus of cells. Through the design of plasmids, we can easily replicate genes that we want to distribute. Designing plasmids largely requires two proteins, restriction enzymes and DNA ligase. Together, they work in a process essentially cutting and pasting genes. Restriction enzymes target the old gene and cut it out of the plasmid at the restriction site. Different restriction enzymes target different sections of DNA, so the enzyme used is dependent upon where in the DNA sequence the cut needs to be made. Usually the enzyme cuts the DNA so that there is a short overhang on each end, which makes them easier to attach to other pieces of DNA. After the section is removed, DNA ligase is needed to attach to the target or interest gene to the one to be replicated. These can be specifically designed genes or genes found in plasmids in different parts of the species. DNA ligase pastes the target gene into the plasmid by joining DNA with matching ends, creating an unbroken molecule or plasmid. Since plasmids are easily replicated, the gene can now be easily distributed. This is an example using EcoRI, a specific type of restriction enzyme. Here we have a target gene and the plasmid vector. The target gene has two EcoRI sites near its ends, and the plasmid has one. The EcoRI then cuts both ends of the target gene in the same pattern, then does the same to the site at the plasmid. Finally, the DNA ligase combines the target gene with the plasmid. The overhang ends stick together with complementary base pairing, A's and T's and G's and C's. Once the completed plasmid has been formed, it can then be introduced into bacteria such as harmless E. coli in a process called transformation. During transformation, bacteria is usually heated to high temperatures, which encourages them to take up new DNA. As the plasmid enters the DNA, they continue replicating so that they can be widely distributed. I hope you have learned a little about how plasmids are used in synthetic biology in this video. Thank you for watching from our Del Norte iGen.